Good afternoon, everyone. Thank you for joining us today. I'd like to welcome you to the Student Chapter Organized 787th monthly meeting for the Eastern PA branch of ASM. My name is Michelle Kutzler, and I serve as the Student Chapter Faculty Advisor as well as President-elect. And I'm also Associate Professor of Medicine and Microbiology and Immunology at Drexel University College of Medicine. So before I turn the meeting over to our student chapter officers, I'd like to review just a few logistics just to get us started on how we're going to run the Zoom today. So the schedule is we will first have our 4 student talks from 4 to about 5:10, and then we're going to take a quick 5-minute break before our keynote seminar speaker, Dr. Shantanu Bhatt, who will start that around 5:15. And when you're participating in the meeting, we'd like for you to utilize the chat feature. So if you have a question, please type it in the chat for our speakers. We will have a few minutes at the end of each talk so that we can answer questions, and our student moderators will get to your questions in the chat. You can also click on the participants tab and raise your hand if you'd like to ask the question yourself. So our student moderators will be looking for either option for you to be able to ask your questions today. I'd like to take a moment just to talk about ASM and what a wonderful professional society it is, and that there's a great community network, also professional development and science and cutting edge science. So just to remind everyone on the call, if you're not yet an ASM member, you can renew your ASM membership for 2021. The new membership year begins this October. And there's a great deal of online resources and on-demand sessions that are available through ASM Microbe Online. So please spend some time at asm.org to look at all the great resources, including these keynote addresses that are listed here. A really great opportunity as an ASM member to see some great scientific talks. I'd also like to remind colleagues about the ASM Awards and Prize Program. So this program is managed by the American Academy of Microbiology. This is the honorific leadership group within ASM. So the mission of this academy is to recognize microbiologists for their outstanding contributions to the microbial sciences and to provide expertise in the service of science to the public. So being nominated for one of these awards and receiving one of these awards is quite an honor for any microbiologist. And it really helps to advance the careers of our early stage career scientists, as well as bringing recognition and visibility to our institutions. So there are awards for exemplary teaching, research and environmental science, as well as basic and applied and biotechnology research, as well as clinical microbiology, from early career all the way to lifetime achievement. So please take a moment to recognize your colleagues through the ASM Awards program that is open. Nominations are due by June 28th, and the awardees will be announced this September 2021. So with that, I'd like to introduce and turn over the presentation and our wonderful speakers today to the Eastern PA branch student chapter officers. So I'm going to be introducing Matt Bell, Kayla Sakaris from the College of Medicine at Drexel, our Vice President, Marissa Egan from the University of Pennsylvania, our Secretary, Caitlin Grando, the Lewis Katz School of Medicine at Temple University, and our treasurer, who we're looking for another student who's interested in serving to let us know. We do have an opening for treasurer. And I'd also like to take the opportunity to introduce and welcome Dr. Sunny Shin, Associate Professor of Microbiology at the University of Pennsylvania. She will be taking over as our Student Chapter Advisor this July. So welcome, Dr. Shin, and thank you so much for your service. And with that, I'm going to turn it over to our student officers, President Matt and Kayla. So Matt, if you could unmute, thank you.
My bad. Sorry, guys. So I'm Matt Bell. Hello, everybody. And along with Kayla, I am co-president of the student chapter. I'd like to thank everybody on the student chapter team, including Kayla, Marissa, and Caitlin, along with our faculty advisors, Dr. Putzler and Dr. Shin, for helping to organize this meeting. In addition to organizing this May monthly meeting, the student officers also helped to organize the annual Philadelphia Infection and Immunity Forum. And we're hoping to have that in person this year. So we're very excited about that and getting back to meeting in person with everybody, as well as organizing the Philadelphia Infection and Immunity Forum. We also host events such as judging at the George Washington Carver Science Fair and the Science After Hours at Franklin Institute. And I'd also like to highlight that we are in search, like Dr. said, of a treasurer. So if you are potentially interested in joining student officers, please reach out to one of our members. The contact information, email address for the student chapter is down in the lower left-hand corner. And with that, I will turn it over to Kayla.
Hi, everyone. Thank you so much for that, Matt and Dr. Kutzler. So what I quickly wanted to say is to please join our branch. It is for free, and how you can do that is by simply going through to the website for our local branch, and you can click there and do kind of an e-sign up, where really it's free for students. It's actually a wonderful opportunity since our branch, especially the student board, is involved in a lot of different events. So joining EPAASM allows you to be able to interact with a lot of invited speakers before the pandemic and hopefully soon after. You can actually have lunch with them, and it's a great networking opportunity, and a lot of good can come out of it. And there's also the ASM Young Ambassador Program, which also hosts a number of different events towards students and early career scientists. So an example of that is the upcoming webinar about how to launch your career in biotech industry. So please feel free to reach out to us. Our contact email is down below. It's EPAASM_students@gmail.com for those of you where the screen may have been cut off. And hopefully I look forward to hearing from you all soon. So that's it for me for some stuff, and we can start introducing the student presenters. Okay. So I'll be doing the first introduction, and so I'm more than happy to present our first student speaker for tonight. It is Mr. Alexander Smith. He is a PhD candidate from Dr. Zakular's lab over at University of Pennsylvania, Perelman School of Medicine. And the title of his talk is Microbial Cooperation Enhances Clostridium difficile Pathogenesis. Thank you so much, Alex, and I look forward to hearing your talk.
Okay, thank you. And thank you to the organizers for giving me the opportunity to speak today. Like Kayla said, my name is Alex Smith. I'm a third-year PhD candidate in Joe Zakular's lab at UPenn and CHOP. And the title of my talk is Microbial Cooperation Enhances C. difficile Pathogenesis. And so the pathogen that we study in the Zakular lab is Clostridioides difficile. It's a gram-positive spore-forming obligate anaerobe and causes disease through the action of its two potent exotoxins. And the CDC has classified it as an urgent pathogen, given that it causes hundreds of thousands of hospitalizations every year, tens of thousands of deaths, and billions of dollars of excess healthcare costs. And so of particular interest to our lab is how CDI patients, C. diff infection patients, can suffer from a wide spectrum of disease severity. And so anything from asymptomatic colonization, primarily seen in infants and in a low population of adult patients, through mild diarrhea, pseudomembranous colitis, toxic megacolon, and possibly death, with a high chance of recurrence if you clear the primary infection. And so a lot of studies have been done to try to assess the effect of the microbiota on antagonizing C. diff infection. And so C. diff enters a very rich dynamic polymicrobial environment. And so, many bacterial taxa have been identified that possibly antagonize the virulence of C. diff. However, of particular interest to me are members of the microbiota that actually enhance C. diff infection. And so less work has been done in this area, but the studies that have been done, both in our lab and others, have identified several bacterial taxa that tend to be positively correlated with C. diff infection and even severe C. diff infection. And one of those genera that I particularly study is Enterococcus. And so Enterococcus is a normal human gut commensal, but it also can cause disease as an opportunistic pathogen in patients with risk factors that significantly overlap with C. diff infection. And so clinical studies that we've performed in two different cohorts, namely children with C. diff infection versus inpatient controls at Vanderbilt University, shown in the gold and black, as well as children with IBD and C. diff versus healthy controls from here at the Children's Hospital, show elevated levels of Enterococcus in patients with C. diff infection compared to patients without C. diff infection. And actually in our clinical cohort from CHOP, C. diff is actually positively correlated with enterococcal burdens. So the more Enterococcus these patients have, or I should say the more C. diff these patients have, the higher Enterococcus burdens we tend to see in these patients as well. And so I wanted to, the primary focus of my project is to investigate whether this correlation that we see in patients actually represents kind of a relationship or an interspecies interaction between these two bugs. And so in order to kind of get at this question, we first turn to our in vivo model of C. diff infection, where mice are sensitized with antibiotics before infection with C. diff. And so if you sensitize the mice with a broad spectrum antibiotic, cefoperazone, and then infect with C. diff and track burdens of C. diff and Enterococcus over time, you can see that Enterococcus burdens are elevated during antibiotic treatment, but then really skyrocket as soon as you infect with C. diff. And so if you knock down the commensal Enterococcus population by treating with vancomycin, you actually see that Enterococcus seems to support successful C. diff colonization. And so if you knock down Enterococcus in red to below the level of detection prior to C. diff infection, you actually get a delay of colonization of C. diff by about a day relative to in the cefoperazone model alone. And then this phenotype can actually be complemented by adding back Enterococcus. So this is just showing the C. diff and commensal Enterococcus burdens under the Ceph column on the left of this figure. And these are the levels kind of shown here at the beginning of infection. And then in our Ceph and vancomycin model, if you knock down the commensal Enterococcus population, you get a significant reduction of C. diff colonization on day 1. But in this same model, if you gavage in Enterococcus, exogenous Enterococcus, at the same time as C. diff infection, you can actually recover C. diff colonization on day 1 to levels seen in the cefoperazone only model. And furthermore, interestingly, in the mice that were given exogenous Enterococcus, they tended to have statistically higher levels of the C. difficile toxins in their stool compared to the C. diff only infected group, implying that there's an increase in C. difficile virulence corresponding to elevated levels of Enterococcus in the gut. And so the question becomes, how does Enterococcus affect C. difficile virulence? And so for this, we kind of left the in vivo model and went in vitro to kind of understand the effect that Enterococcus could be having, particularly with soluble factors. And so for this, I performed a conditioned media assay where I grew Enterococcus, and then I grew C. diff in the spent supernatants of the Enterococcus cultures, and then assessed toxin production through an ELISA, which measures protein levels, and through a virocell cytotoxicity assay, which measures functional toxin activity. And both of these assays revealed that when C. diff is grown in the conditioned media of both a vancomycin sensitive strain of Enterococcus OG1RF and a vancomycin resistant strain of Enterococcus V583, we see elevated levels of C. diff toxin production compared to when C. diff is grown by itself, indicating that Enterococcus is promoting C. difficile virulence through a contact-independent mechanism. And so to further understand the metabolism behind these changes, and it's not shown here, but we performed a co-culture RNA sequencing experiment where we compare the transcriptomics of C. diff and Enterococcus when grown together versus when they are grown separately. And so we've got a lot of results from this experiment, more than I'm going to show today. But one of the biggest changes that we saw was that when C. diff is grown with Enterococcus, it shifts its metabolism toward primarily amino acid fermentation, which indicated to us that amino acid crosstalk and amino acid metabolism was playing a central role in this interspecies interaction. And so to understand that crosstalk, we wanted to perform spatial metabolomics to understand what could be happening here. So on the left here, I'm showing what these two organisms look like when they're co-cultured on solid phase. And so Enterococcus is in the middle, and then compared to when it's grown by itself, C. diff undergoes this morphotype switch to this kind of dendritic blooming phenotype, which is kind of its own interesting story of that mechanism that I will give in another talk someday. But on these co-cultures, we performed something called imaging mass spectrometry, which allowed us to localize the distributions of amino acids across this co-colony. And an interesting pair that really stuck out from this data was arginine and ornithine. And so ornithine is highly enriched at this interface between Enterococcus and C. diff, corresponding to this ring, while arginine is massively depleted in the exact same area. And so if you kind of look into metabolism of Enterococcus with arginine and ornithine, the first thing that comes up is the arginine deaminase pathway. And so this is a method of metabolism that Enterococcus and its relatives can use to metabolize arginine to produce ATP and then export ornithine as a byproduct. And so we performed liquid metabolomics on our Enterococcus cultures to visualize this. And so these are the levels of arginine and ornithine in just our media. When you grow Enterococcus, you can see a consumption of arginine and a concomitant production of ornithine. And if you knock out this antiporter ARC-D responsible for exchanging these two amino acids, you can completely abrogate this consumption pattern in Enterococcus. And so if you use this Enterococcus ARC-D mutant in the conditioned media assay that I showed previously, you actually get a complete abrogation of this toxin increase phenotype. So when C. diff is grown in the conditioned media of the ARC-D mutant, compared to wild type Enterococcus, toxin production goes down to levels seen when C. diff is just grown by itself. And this indicates one of two things. Either arginine depletion is driving an increase in C. diff toxin production, or ornithine production is the amino acid that C. diff is sensing to increase its toxin production. And to assay this, I simply added back arginine to the conditioned media. And interestingly, so adding back arginine to the Enterococcus conditioned media phenocopied the ARC-D mutant. And this indicates that it's in fact arginine depletion that's increasing C. difficile toxin production, which is in line with previous literature on C. diff amino acid metabolism. And so I wanted to take this back into an in vivo setting to kind of see what kind of an effect this would have on a real infection. And so turning back to our cephalosporin model of C. diff infection, at the conclusion of antibiotic treatment, but before infection, I replaced half of the mice, their drinking water, with water that had been supplemented with L-arginine, and then tracked infection over time. And so here you see that the C. diff burdens actually over the course of infection between these two groups doesn't really change. They're not significantly different. However, the C. diff alone group tended to lose a lot more weight over the course of infection than the mice that were supplemented with arginine. Along the same lines, if you track a clinical sickness score, which is a composite of several disease severity metrics, the C. difficile mice tended to do worse in terms of both stool composition and behavior relative to the mice that were supplemented with arginine. And finally, the mice that were supplemented with arginine actually had lower levels of toxin, C. difficile toxin, in their stool, despite the fact that there was no difference in C. difficile burdens in either set of mice. And so this is indicating that this is this in vitro phenotype that we see correlates well to in vivo, where adding back arginine actually lowers toxin production by C. diff. And so kind of future directions with this project, I told you a lot about what arginine does, but what about the effect of ornithine? Is there an effect? And so this was actually data that came from our RNA sequencing run. We were able to run that data through a metabolic model to kind of in silico predict features of C. difficile growth when grown with Enterococcus. And one of the predictions was that its fitness actually increased in the presence of Enterococcus. And that kind of aligned with a predicted increase in the uptake of ornithine. And so the data I'm showing here fits well with that model. This is actually on the left is clinical data from the cohort of children that I showed in the beginning. And actually C. difficile burdens track positively with ornithine abundance in the stool, possibly indicating some kind of biomarker. Additionally, if you infect germ-free mice with either C. diff alone or C. diff plus Enterococcus, there's higher levels of ornithine in the stool of mice where Enterococcus is present. And so we believe that ornithine could be having an increased effect on C. difficile fitness, that C. diff could be using ornithine in its amino acid fermentation mechanism in order to produce energy and kind of gain a competitive advantage. And so this altogether supports a model where Enterococcus is simultaneously restricting arginine and producing ornithine, which provides signals to increase C. difficile toxin production and possibly increases fitness in kind of a positive feedback loop to increase virulence and disease severity. And with that, I would just like to thank my lab as well as all of our collaborators and for the organizers for this opportunity to speak. Thank you.
Thank you so much, Alex. That was a wonderful talk. Does anyone have any questions?
Hi.
You can post your question in the chat if that is easier for you, if you're having some sound issues.
Okay, Dr. Kutzer, do you want to go ahead with your question?
Okay, I can ask the first question. Great job, Alex. Really, really great study.
Thank you.
Is related to the clinical cohort, the stool samples from the clinical patients. They're in children, so they're under 18, and then the animal model is adult mice. So is there an age component that you're going to be investigating? Is there differences between young versus old?
That's an interesting question. That's something that I think we're definitely going to explore because we're working right now to get an adult cohort, clinical cohort, to kind of look at these same effects in. And actually the lab is just kind of up and running with playing with kind of age-related infections in mice and looking at younger mouse cohorts, mouse infections. And so that's definitely something that we can study. And I would definitely be interested in kind of dietary changes throughout life. Because I think like the diet component and the metabolism is something that I'm really kind of leaning into as this story has progressed. So I definitely think that would be something that we're going to kind of get into in the near future.
So Kayla, do you want to read the question from the chat? Lucy posted that. Yeah.
Yes. Actually, it seems like we have two questions. So I'll go with Lucy first, to kind of keep the order and then proceed to the next one. So the first question is, does the ornithine and arginine relate to the changes in the urea cycle?
So yes, we definitely think that there's a relationship with the host, especially with the urea cycle. And so I didn't have time to show it now, but we have a lot of that imaging mass spec data from infected mouse Zika. And something that has shown is that it actually seems that the predominance of arginine that we find in the lumen during infection seems to be tissue derived instead of dietary derived. And so there's definitely a relationship with arginine release from the host, possibly due to the damage from the C. difficile toxins. I have not really examined the host side yet, but especially now that we've kind of gotten into, or given how tightly linked arginine is with both the immune system and with general, eukaryotic metabolism, that's definitely an angle we're going to pursue.
Okay. Well, we have one quick question left, and after that, we're going to have to move on. If anybody has any questions, feel free to keep putting them in the chat. So this last question is arginine is used by macrophages to make nitric oxide. Is there a link to the host defense?
Yeah, so this is kind of in a similar vein. And so I definitely think there's a role being played by the immune system with arginine release into the tissues. And so that's kind of a future direction for us to see both where the arginine is coming from, because I think, arginine consumption and release from immune cells at the epithelium, especially during like C. diff infection, could be a major source of the interplay here that we're seeing.
Wonderful, thank you. All right, so unfortunately that is the end for the time we have for questions for Alex, but please feel free to keep going in the chat.
Thank you, everyone.
Mm-hmm. All right, I will be introducing our next speaker. So our next speaker is Beatrice Herman. She is a PhD candidate in the lab of Dr. Igor Brodsky at the University of Pennsylvania Perelman School of Medicine. And she'll be presenting her talk on defining the bacterial and host factors responsible for caspase-1,11 independent cell death during infection by Salmonella, Cerevar, and Enteritis. If you feel comfortable answering that in the chat, maybe.
Yeah, sure. Thank you. Sorry.
Okay, great. Thanks, Beatrice, for that amazing talk. So next up, I'm going to introduce Matt Bell. So Matt is one of our co-presidents, and he's also a third-year PhD candidate in Dr. Kutzler's lab at Drexel. Today, he will be presenting his talk entitled Age-Associated Defects in the Adaptive Immune Response to C. diff Infection Impair the Development of a Protective Immune Response in the Elderly. Thanks so much, Matt.
Great, thank you, Marissa. So there we go. So the first big question, obviously, is what is C. diff? And I know we've already talked about this a little bit today, so I'll keep this relatively brief. C. diff is a causative agent of C. difficile infection, CDI. It is gram-positive and spore-forming, and it has a relatively large impact on the United States and our healthcare system because we know that CDI is both deadly and costly. There's around 200,000 cases and 13,000 deaths per year in the United States, and around 20% of individuals who have a primary case of CDI are going to wind up with recurrent infections as well. CDI also adds up to 12 days in the hospital and around $27,000 per case in direct costs. And in terms of the annual cost of CDI, it's around $1.5 billion. And so you might be wondering, what are some of the risk factors for getting CDI? One of the leading risk factors for CDI is advanced age. And we know that around 70 to 80% of CDI cases are going to occur in those individuals who are 65 years or older. And we also know that over 80% of deaths are also going to occur in those who are 65 plus. And so then the really important question becomes, what are some of these underlying causes that lead to increased risk of CDI in the elderly? And the answer to that question is that there are a number of different possible contributing factors to the increased risk in the elderly, including increased polypharmacy, comorbidities, increased healthcare exposure. And it's also true that as we age, our immune response is going to wane. In particular, the adaptive immune response is affected, and the humoral response in particular, we know with the aging host, there's a decrease in antibody titer production, high affinity antibodies, and also isotype switching. And we believe that this, along with others, are contributing to the poor outcomes that we see in elderly CDI patients. Now, one of the reasons that we believe that the humoral response plays a really important role in preventing the pathology of C. diff is because C. diff has two major pathogenic factors, toxin A and toxin B. These toxins are primarily going to work on the intestinal epithelium, where they lead to the breakdown of tight junctions through cell death. And the neutralization of these toxins can help to prevent C. difficile-associated disease for people who are infected with C. diff. Now, we believe that age-related deficits in the humoral response are going to lead to the increased risk that we see in C. difficile-associated disease. And so the question that we had is the humoral response impaired to CDI in advanced age? And to answer that question, we have run challenge experiments where we challenged both young and aged mice. Following antibiotic exposure, we challenged them with either a sublethal or lethal dosage of C. difficile spores, and then monitored for morbidity and mortality. We also collected sera from the surviving mice. And what we found is that there is a noticeable increase in the risk of mortality in aged mice, both for the sublethal and lethal infections. So for the sublethal infection, as expected, 100% survival in the young mice in blue, around 30% mortality in the aged mice. For the lethal infection, it's even more distinct, where you have only 10% loss in young adult mice, but 100% fatality by day 4 in these aged mice. Again, highlighting the fact that there's an increased risk of mortality with advancing age, something that we also see in elderly CDI patients. Now, we also measured the humoral response in the surviving mice that survived the sublethal challenge. And what we found is that there's a decreased humoral response in the surviving aged mice. And so the aged mice in red, toxin A responses, toxin B responses, you can see there's a significant decline in IgM, IgG, and IgA in aged mice against toxin A, and an IgM, IgG, and a trend towards IgA in toxin B. And so this age-related decline in humoral response to CDI, we believe, is a contributing factor to the increased susceptibility to recurrent CDI that we see with advancing age. Now, we know that the humoral response to a primary infection with advanced age is deficient, but the next question we had was, can we develop a vaccine that can protect against C. difficile-associated disease in the elderly? And this is really important because there's currently no licensed vaccine for C. diff. So to that end, we developed a DNA vaccine targeting the toxins A and B receptor binding domains. And the receptor binding domains are really important targets for a vaccine against C. difficile-associated disease, because we know that neutralization of the toxin RBDs prevents endocytosis and therefore the cytotoxicity of the toxins. It's also true that the RBD sequence is well conserved between strains of C. diff, and so this can provide cross-reactive protection between the different strains. And so to test if we could get a strong humoral response to vaccination in aged mice, we vaccinated both young and aged mice with RBD expressing plasmids. And we gave 2 doses over the course of 26 days and then stacked the mice to look at the humoral response. And ultimately, what we found is that the humoral response to vaccination in aged mice against both toxin A and toxin B is significantly declined compared to the young mice that were vaccinated, suggesting that just as we saw with the primary infection in aged mice, there's also deficits in the humoral response to vaccination in aged mice as well. So one of the prime ways that we can really boost the vaccine response is through the use of adjuvants. And currently, the Kutzler and Hadad lab are investigating a potential adjuvant called ADA1. ADA1 is an enzyme that's known to play important roles in immune synapse formation, as well as TFH differentiation and function, and T cell proliferation. And so we believe that ADA1 can play a potential role in boosting this humoral response that's deficient in aged mice and possibly lead to a protective immune response. And so we've cloned ADA1 into a vector for delivery as a molecular adjuvant called pADA1, and to see if it does in fact lead to a boosted humoral response in aged mice, we vaccinated both young mice and then aged mice with the addition of ADA1. And we again gave 2 doses and then stacked the mice to look for the humoral response. And what we found is that with the addition of pADA1, it restores antitoxin A and B humoral response, in aged mice versus young adult mice, both in terms of IgG against toxin A and IgG against toxin B, suggesting that ADA1 can work as an adjuvant to boost the humoral response in aged mice. And so to summarize what we've shown thus far in some of our studies, we've shown that the age response to infection and vaccination, we see a decrease in antibody titers, isotype switching capabilities, and we're currently running neutralization assays to look if there is also a decrease in neutralization ability with advancing age, which we suspect leads to greater susceptibility. And so all of this combined together leads to poor outcomes that we see with advancing age. What we've also shown is that the age response to vaccination with the addition of PADA1 leads to a restoration of the humoral response, at least in terms of increasing antibody titers, isotype switching capability, and we're currently looking at neutralization ability. And our hope is that it will also increase neutralization and therefore lead to a protective response against C. difficile associated disease. So quick summary and future directions. Like I said, the immune response to infection and vaccination is defective in advanced age. ADA1 shows promise as an adjuvant, especially in the context of an aged immune response. And some of the future directions that we have looking at PADA1 as an adjuvant is to look at neutralization abilities, but also look at some of the mechanisms that we believe PADA1 is working through, such as the antigen-specific TFH and B cell responses. We believe that ADA1 is boosting these responses. And we're also going to run challenge experiments following vaccination to show that it does in fact induce a protective response. And so I'd like to end with acknowledgements, thanking everybody in Kutzler and Dodd Lab who played a really essential role in making these studies possible, as well as all of my committee members. And I'd like to thank everybody else involved in these studies. Thank you, everybody, for listening, and I'll end with questions.
Thanks so much, Matt. That was a great talk. So we have one question in the chat from Dr. Eisenstein. She asked, is nanogram levels of antibody protective? She said she would not think so.
Yeah, that's a great question. So one important thing to mention here is that these are all log scale. So they're pretty high antibody titers. And that's one thing that we'll be looking at with the neutralization assay as well, and then with the future challenges and seeing if these responses are actually protective. That's a great question.
Awesome. Thanks so much. I think for the sake of time, if anyone else has any questions, just please put them in the chat for Matt, and Matt, if you wouldn't mind answering them, and then I'll turn it over to you to introduce Ellen, but great job again.
All right. So our next speaker is Ellen White. So please allow me to introduce her. She's our final student speaker, and she's an MD PhD candidate in the Grice Lab at the University of Pennsylvania Perelman School of Medicine. And she will be presenting her work on microbial-based mechanisms of accelerated wound healing. So with that, I'll turn it over to Ellen.
I apologize. I was having some problems with unmuting myself. I'll reshare my screen. So I want to thank the organizers for inviting me to speak today. And I'm excited to talk about my thesis work studying the influence of the skin microbiome on wound healing. In the Grice Lab, we study the skin microbiota, which is the collection of microorganisms at the skin barrier surface. When wounding occurs, there is breach of the barrier, which allows contamination of this resident skin microbiota into the wound bed. After contamination occurs, there's a spectrum of wound bioburden, which really we think of as a continuum of wound infection. The effects of bacterial bioburden in the setting of avert infection is obvious, where high bacterial bioburden leads to poor outcomes like amputation and osteomyelitis. What is less obvious is the colonization effects, where the bacterial bioburden is relatively low. We are interested in how microbial colonization affects healing outcomes because at this stage, the skin microbiome is an important component of the wound environment, and offers a novel therapeutic target for improved healing outcomes. There is a critical need to improve healing outcomes because of the burden of chronic wounds. There are 5 to 7 million episodes of non-healing cutaneous wounds each year in the US, and these contribute 20 billion to the annual healthcare cost. A common example of chronic wounds that we study in the Grice lab are diabetic foot ulcers, or DFUs. So in order to begin to answer the question of how microbial colonization affects healing outcomes, our lab profiled the microbial community of DFUs. To do this, in the study that was foundational for my project, we collected longitudinal samples from non-infected DFUs of 100 patients. We then performed metagenomic shotgun sequencing on these samples. The sequencing experiment identified that the top 3 most abundant bacterial species in our cohort were predicted skin pathogens such as Staph aureus, as well as the skin commensal Corynebacterium striatum. We are surprised to find that the 4th most abundant species in this cohort was Alcaligenes faecalis, and we were able to collect 44 isolates of this species from our cohort. This is a surprising finding because Alcaligenes is not typically studied within the skin microbiome field and is often dismissed as an environmental contaminant. However, it's not just in our cohort that Alcaligenes is isolated from wounds. It's also been isolated from other types of chronic wounds like pressure ulcers. And in a recent survey of the skin microbiome of sickle cell disease leg ulcers, Alcaligenes was also a prominent component. Because how abundant Alcaligenes is in these wound cohorts, and because of how little research has been done on Alcaligenes in the context of wounds, we wanted to ask what the effect of Alcaligenes is on the host wound healing response. One of the first studies we did to ask this question is monocolonize the wounds in a diabetic mouse model with the bacterial strains. We were surprised to find that treatment of these wounds with Alcaligenes led to accelerated wound closure compared to the untreated control, and this phenotype was especially prominent at early time points in wound healing. This is in direct contrast to treatment with Staph aureus, which slowed wound closure. So my project is really trying to understand the mechanisms of Alcaligenes-mediated wound healing. To address this question, my project has 2 arms. First is understanding the Alcaligenes-mediated host responses that mediate this accelerated wound healing. And the second component is understanding the microbial determinant of accelerated healing. I will first focus on these host responses to Alcaligenes. When we think of the host wound healing response, it's a complex process with many overlapping phases. For my project, I'm focusing on the early phases of the inflammatory and proliferative stages. And common to both of these phases is the process known as re-epithelialization. This is a really critical stage of wound healing, and in fact, most chronic wounds do not progress past this point. In addition, these early stages are when I observed the strongest phenotype of Alcaligenes-mediated healing, which I previously highlighted. So one process that's necessary for re-epithelialization is inflammatory cytokine signaling by keratinocytes. So in order to pin down the mechanism, one of the first experiments we did was to ask if Alcaligenes can induce this inflammatory cytokine signaling keratinocytes. To test this, we treat primary keratinocytes that have been scratched with cell-free bacterial conditioned media, and then we can profile the inflammatory cytokine response with the multiplexed ELISA. In this experiment, we found that Alcaligenes induced robust cytokine response in multiple inflammatory cytokines, and this response was not seen after treatment with Corynebacterium conditioned media or with 2 isolates of Staph aureus. One cytokine that I just want to briefly highlight is IL-6, which I use as a downstream readout in future experiments because this cytokine has been shown to induce a pro-epithelialization phenotype in keratinocytes. In addition to this necessary cytokine signaling, Another activity of keratinocytes that is necessary for reepithelialization is keratinocyte migration and proliferation. I can test the effect of alkylagenase conditioned media on keratinocyte migration through several in vitro assays. I've just highlighted one here for simplicity. This is the trans-well migration assay, in which bacterial conditioned media is plated in the bottom well. In the top well, primary keratinocytes are plated in serum-free media. We can then quantify the amount of keratinocytes that migrate towards the bacterial conditioned media. In this type of experiment, I have found that alkylagenase induces keratinocyte migration, and this is not found after treatment with Corynebacterium conditioned media or Staph aureus conditioned media. In terms of keratinocyte proliferation, we can analyze the leading edge of the wound, which is the path of migrating keratinocytes. We can quantify keratinocyte proliferation through methods like immunofluorescence, which I've shown here, in which we use K14 as an epithelialization marker for keratinocytes and EDU as a marker for proliferating cells. Here you can see that treatment with alkylagenase leads to increased proliferation of keratinocytes at the leading edge. And again, this phenotype was most prominent at these early time points of healing. So my host work thus far has demonstrated that alkylagenase can increase the reepithelialization potential of the host. I'm also interested in determining how other cell types that are important during wound healing are responsive to alkylagenase treatment. One method I am performing in order to test these host responses is performing RNA sequencing on wound tissue that has been treated with alkylagenase. I now want to focus on identifying the microbial determinant of accelerated healing. And one approach I am taking to identify this bacterial determinant is a comparative genomic approach, in which I'll identify the genetic determinant of the pro-healing response. I'm able to take advantage of this kind of approach because we have this collection of 44 clinical isolates. I have already sequenced this collection of isolates, assembled the whole genomes, and annotated these genomes. So the goal of this type of approach is to associate a specific genotype with the pro-healing phenotype. The collection of isolates has a large amount of genetic diversity, which I've highlighted here with the phylogenetic tree. And I have selected a panel of 10 isolates that I've highlighted in yellow for downstream phenotypic assays. And these yellow isolates, you can see, are representative of the genetic diversity of the collection of isolates. I then hope to integrate this genetic screen with phenotypic screens for the pro-healing response through high-throughput cytokine screens and wound healing assays. So with this type of genetic approach, it's helpful to have a hypothesis of what kind of bacterial product I'm interested in. I started with the hypothesis that the active molecule is a secreted factor rather than a cell surface associated molecule, given that all of my previous findings were done with cell-free conditioned media. So to test this hypothesis, I can compare the cytokine response of scratch keratinocytes of keratinocytes treated with heat-killed bacteria compared to those treated with conditioned media. With this experiment, I have found that at several doses of heat-killed bacteria, I do not get a robust cytokine response in keratinocytes, and this is in comparison to alkylagenase conditioned media. Another way to characterize this bacterial secreted factor is to perform size fractionation on the conditioned media to try to pin down what size of factor I'm looking at. So one of the first experiments I've done is size fractionation with a 3 kilodalton filter. After this fractionation, I have a larger fraction and a smaller fraction, and I can treat scratch keratinocytes with these two fractions. From this initial experiment, I have seen that the larger than 3 kilodalton fraction has higher activity and similar to that of conditioned media compared to the smaller than 3 kilodalton fraction. So from these preliminary experiments, I hypothesized that the bacterial active factor is likely a large secreted molecule. So in terms of future directions for identifying this microbial determinant of accelerated healing, I can do further characterization of the conditioned media with experiments like peptidase treatment. And in terms of further progressing the comparative genomic approach, I am working on performing a genomic multiple alignment of all of the alkylagenase genomes, and again, performing these phenotypic screens on my selected panel of isolates. So completion of both arms of my project will allow me to identify both host and bacterial mediated mechanisms of improved healing, which will allow us to exploit these interactions to develop novel microbiota-based therapies. So in conclusion, I want to thank my lab, who's been very helpful with the development of this project, our collaborator, Sue Gardner, who helped was critical for the original DFU cohort study, as well as my funding sources. And I'm happy to take any questions.
Great. Thank you, Ellen, for your talk. So it looks like we have 2 questions. So the first one is from Igor. So he says, this is fascinating, Ellen. A few years ago, collagenase was identified to disseminate systemically in animals with disrupted intestinal barrier defenses. What is the thought on how collagenase might get to the wound site and skin wounds in diabetic patients? Do diabetic individuals have defect in intestinal barriers or expand alkalogenase in their gut microbiota? Or is alkalogenase a low abundance bacterium in the skin that expands as a result of an inflammatory environment?
That's a great question. So yeah, that's definitely one thing we're interested in, is how alkalogenase, which is not really thought of a normal component of the skin microbiome, is showing up in many of these wounds. I actually haven't thought of the whole gut-based hypothesis, where I think you're implying that maybe the origin is through their gut. Diabetic individuals, I would believe, might have a mild defect in their intestinal barrier, just given that they have sort of a chronic inflammatory state. I think your second point is sort of more what I think of, where when there's these chronic, where there are wounds, there's disruption of the normal skin flora, which allows the opportunity for these outside bacteria to populate the surface of the wound. So because we think of alkalogenase as environmental bacteria, is it possible that it's coming from environmental surfaces and is now able to thrive in this wound area? That's my hypothesis.
Great, thank you. So there is one more question in the chat, but I think for time's sake, we'll end it there. And if you want to respond in chat as well, then great.
Yep, and I guess now we can take a quick break until 5:15. So let's take a 7-minute break, and then we can resume, and Dr. Bott will give us his keynote seminar. So we'll see everyone at 5:15. Thank you. All right, since it's 5:15, I think we'll get started. Hopefully everyone is here and coming back. So I'll start by introducing Dr. Bott. So hi, everyone. It's my pleasure to introduce Dr. Shantanu Bott as our student invited speaker today. I had the honor and the privilege of being mentored by Dr. Bott during my undergraduate career at St. Joseph's University. Dr. Bott is an extraordinary scientist, a dedicated mentor, and an incredible teacher. Dr. Bott graduated from Denison University, and then he pursued his PhD in microbiology and molecular genetics from Emory University. During his PhD, he studied how the RNA binding protein CSRA regulates the virulence of enteropathogenic E. coli, or EPEC. After his PhD, Dr. Bott did a postdoc at the NIH, working on investigating the regulatory roles of small RNAs and small proteins in E. coli. After his postdoc, Dr. Bott became a professor in the biology department at St. Joseph's University. Dr. Bott has made significant contributions to the field of microbiology, as his lab has identified numerous small RNAs that regulate the virulence of pathogenic strains of E. coli like EPEC. Moreover, Dr. Bott has mentored countless students in his research lab and his classroom. He's been recognized as a dedicated professor who cares deeply about teaching and mentoring. Most recently, he was awarded a Faculty Merit Award at St. Joe's, which is a well-deserved accomplishment for Dr. Bott. Personally, Dr. Bott has inspired me every day since my first day in his classroom and his lab. He taught me that in science and in life, the joy is in the journey, and I'm forever grateful for Dr. Bott's mentorship. I know I would not be here without him today. And I know that I'm not alone in my admiration for Dr. Bott. Dr. Bott is revered by his students and his colleagues alike. So without further ado, please join me in welcoming my scientific hero, Dr. Bott.
Marissa, thank you so much for that wonderful introduction. I don't think I have ever received an introduction like that, and I don't think I'm deserving of it, but thank you.
You're very deserving, Dr. Bott. Thank you for being here.
So I would really like to thank the student members, as well as the faculty in charge of the EPAASM, because this is truly an honor and a privilege to be able to speak to the next generation of budding scientists, as well as the present generation, I believe. And so I would really like to thank the student officers, Marissa, Alex, Kayla, and Caitlin. Was that right? I may have got it wrong.
I think Matt, but yep.
Matt, sorry. Yes,
good.
Matt. Thank
Thanks,
you so
Dr.
much for the invitation. Okay, so let's see. Marissa, can you see my screen?
Yep, it looks great, Dr. Bott.
Okay, so all right, folks. So today I am going to be talking to you about the work that is done in my lab. It's basically on the roles of HFQ and HFQ-dependent small RNAs in the virulence of these two pathogens, enteropathogenic E. coli, and this recently emerging pathogen called Escherichia albertii. Now, before I start, I do want to say that the overwhelming majority of the data that I'm going to be showing you, it's conducted by undergraduate students. We don't have a PhD program, so I'm primarily reliant on the brilliant undergraduate students like Marissa that have come through my lab. All right, so here's a brief overview of how I intend to present my research. So I'll give you a brief background on E. coli, the non-pathogenic kind that you're all very familiar with, as well as I'll specifically be talking about this particular pathogenic kind of E. coli, enteropathogenic E. coli, although there are many other types of there are many other pathowars of E. coli. I'll then quickly shift gears and talk about the small RNA-dependent regulation of this pathogenicity island in EPEC. I'll be focusing on two small RNAs. One is a magnesium-responsive small RNA called MGRR, and the other is this pair of small RNAs. They're paralogous small RNAs, and the common feature between them is they require this chaperone protein called HFQ to exert their effect. And then towards the latter part of my talk, I will briefly touch upon the recent work that my undergraduate students have done on this emerging pathogen called Escherichia albertii. And I'll culminate the talk by comparing the HFQ-dependent regulatory circuits between EPEC and E. albertii. right, so a brief overview about EPEC. Now, my second favorite pathogen. It belongs to this famous group of pathogens, the attaching and effacing family. This is what the bacteria typically do. They'll adhere to intestinal cells and they'll attach really, really intimately. So here's the bacterium colored in green, and it's forming this kind of protrusion on the surface of, this is a tissue culture cell, but this structure is called a pedestal. Now, enteropathogenic E. coli predominantly, it infects the small intestine, and primarily the infections occur in infants and in developing countries. And there's some records that estimate that there's some mortality rates that are as high as 30%. Now, as I mentioned, it belongs to the attaching and effacing family of pathogens. And this group is characterized by intimate attachment to intestinal cells and the concomitant destruction of the intestinal microvilli. So, as you all know, the microvilli are involved in the absorption of water and nutrients. And so here's what, your normal microvilli are kind of looking like, right? And here, what happens is when EPEC or other attaching and effacing members infect, they completely shave off the microvilli. You don't see any there. And so as expected, your intestinal cells cannot absorb water and nutrients. And there's, accumulation of water in your lumen. This results in liquefaction of fecal matter, which we all, know the clinical term is diarrhea. And because the body cannot absorb water, it results in dehydration. And if it's not controlled, it can even result in the death of an individual. Now, the ability of attaching and effacing pathogens to cause diarrhea and disease is bestowed because of the presence of this pathogenicity island called the Li, or the locus of enterocyte effacement. So the Li it's a cluster of it contains almost 42 genes in there, and it's about 35 to 40 kilobases in size. And it's turned on in response to a lot of conditions that mimic the physiological conditions of the intestinal tract. So a physiological pH of 7.4, intestinal osmolarity, a temperature of 37 degrees Celsius, all of those, they turn on the expression of the Li. And the vast majority of these environmental cues, they exert their effect on this first operon of the Li called Li1. And this codes the master regulator layer. So the Li-encoded regulator is a transcription factor. And when it is expressed, what it does is it synchronizes transcription from the other operons of the Li. So it's, GRIL-RA, Li2, Li3, and you have, ESCD. Now, Li2, Li3, and ESCD, they primarily encode for the structural components of a type 3 secretion system. And the gene, their products, they basically form the proteinaceous components that are embedded in the inner membrane, the peptidoglycan, as well as the outer membrane of the bacterium. Moreover, LAIR, it also turns on the expression of this regulatory operon called GRIL-RA. And GRIL-RA codes for two proteins, GRIL-R and GRIL-A. GRIL-A, A for activator, it is also a transcription factor. And what it does is it actually further reinforces this regulatory circuit. So it further potentiates the effect of LAIR, right? So there's much more amount of LAIR that becomes available. And now LAIR can bind to those promoters for which it has a lower affinity, namely the Li4 promoter. And if you think about it, it kind of makes sense why LAIR targets the Li4 operon only after it's assembled the structural components of the type 3 secretion system that span the bacterial extracytoplasmic space. And the reason is because Li4 encodes for the extracellular components of the type 3 secretion system. It contains the ESPA protein, the monomers, they polymerize together to form this hollow tube that connects the outer membrane of the bacterium to the host membrane. And then ESPB and ESPD, they're kind of like the gatekeepers that get embedded in the host cell membrane. And now you have this continuous type 3 secretion system where the cytoplasm of the bacterium is connected to that of the host cell. And so the bacterium can now traffic contrabands. Now, some of the contrabands that are trafficked are, effectors such as TIR, as well as other type 3 cargo. So these effector molecules are transported through the secretion system. And TIR, this is a very, very important protein because what it does is it gets inserted into the host cell membrane, and then it binds to its ligand that's located on the outer membrane of the bacterium. So here's your tier, and here's the ligand for this protein. This is called intimin for obvious reasons, because it mediates the intimate attachment of the bacterium to the host cell. And so once the bacterium has attached, you see the typical intimate attachment. And now the bacterium essentially forms a permanent tenant in the intestine of the host, where it can just has access to all the nutrients. After the formation of this pedestal-like structure, the bacterium turns off the expression of the type 3 secretion system. And the way it accomplishes this is the second regulator, GRIL R stands for repressor, it can bind to GRIL A and inactivate it, and thereby shutting off the system. So the reason why I got interested in this is because we know that to develop any kind of effective therapies against deep-like infections, you have to have a good molecular understanding of the regulation of the lead. Now, when I started my work as a graduate student at Emory, at that point in time, there wasn't a lot known about the role of small RNAs in regulating the lead. In fact, what was really known about was the proteinaceous factors like transcription factors, as well as some post-transcription factors and post-translational factors, all protein-based systems that control the lead. There was not a single small RNA that had been identified. And in fact, so there's this dearth of knowledge in identifying small RNAs that could potentially regulate the Li, and that's how I got interested in the small RNA world. So a very brief and quick overview about regulatory small RNAs. They're a heterogeneous group of single-stranded ribonucleic acids. They are present in all domains of life, in bacteria, archaea, and eukaryotes. In bacteria, they range in size from about 50 to 500 nucleotides in length. And they are untranslated, so their regulatory effect is inherent in their form as an RNA, not as a protein. And they exert their effects by a diverse array of mechanisms. And the two most common mechanisms are by either they base pair to their target mRNAs, or they can bind to proteins and sequester them. And in these two images, you actually see representative examples of both. So here you have a small RNA shown here in red, and it's base pairing to its target mRNA. And so this interaction can potentially, it can affect, it can inhibit translation from the mRNA. It can even promote the degradation of the mRNA, or it could actually stimulate translation. So base pairing interactions can have these outcomes. Conversely, some small RNAs can also bind to proteins and sequester them, so they can titrate the proteins out. So here in this example, this bubble that you see, this is this protein called CSRA. It'll actually become important in my talk. And here CSRA is binding to its substrate. Let's just call this a messenger RNA. And when you overexpress the small RNA CSRB, what it can do is it can bind to CSRA and sequester it because it mimics the natural substrates of CSRA. And here, just a representative example of a small RNA is shown, and I think this is the small RNA DSRA. Now, most small RNAs that my lab studies, they actually require this chaperone protein called HFQ. And the reason for this is because these small RNAs, they have very limited complementarity to their target. So they need help in order to recognize their potential target, right? So that's where HFQ comes into play. Think of HFQ as a molecular matchmaker. And so what it does is it matches a small RNA to its complementary mRNA target. So HFQ in its functional form, it's made up of 6 identical subunits, and they're all color-coded right here. And it's kind of like, if you think about it, it's kind of like a bagel, right? Just like a bagel has a hole in the center. This is what HFQ is, right? This is the hole in the center. So here, this is a top-down view of HFQ, and this is a sideways view. And so at the top surface and at the bottom surface, HFQ can bind to 2 distinct RNA species, and it uses 2 completely different mechanisms to do that. At the top surface, also called the distal phase, it recognizes those RNAs that are replete with these sequences. So they have an adenine followed by a purine, and then any nucleotide, and an adenine, purine, any nucleotide, so on and so forth. And then from the bottom surface, it actually binds to those RNA species that are U-rich. So kind of like the 3' ends of RNAs, right? They are poly-U-rich. And so HFQ can simultaneously bind to an mRNA at its one face and a small RNA at the other and bring the two together. And if there's complementarity between the two, what can happen is, they can just base pair with each other, right? Complementary base pairing interactions. Okay. So having said that, we became interested in HFQ-dependent small RNAs because one, not a lot was known about the role of small RNAs in regulating virulence in enteropathogenic E. coli. And so that triggered this question. And so here, what I'm showing you is this is just the simplified regulatory architecture of the LEA and how the LEA is turned on, right? Environmental cues, they can affect either grill A or LAIR. And then LAIR can activate transcription from these other transcription units, which results in the formation of these structures called pedestals, where the bacterium is intimately attached to the host cell. So what we did was we first deleted HFQ. And we noticed that in the HFQ mutant, there is an increased expression of grill A, suggesting that HFQ negatively regulates grill A. Consistent with the elevated protein levels, we see that there's an increase in the transcript levels of grill A. So there was almost a 60-fold increase in the grill A transcript in the HFQ mutant compared to the wild type strain. Now, grill A is actually the distal gene that's located in this bicistronic operon grill RA. And so we wanted to see how HFQ affects the expression of grill R, the upstream gene. And similar to what we saw in grill A, the trend was very similar, although not as pronounced as in grill A. In the HFQ deletion strain, grill R transcript levels were also elevated. And so based on this, one would predict that if in the HFQ mutant you have increased grill A, you would predict to see increased levels of LAIR, and likewise you'd see increased expression from all these other operons. And so to test that, we looked at the expression of TIR and ESBB in the HFQ mutant. Now, as I mentioned, TIR is that receptor that the bacterium injects into the host cell, right, which will bind to its ligand on the surface of the bacterium. And ESBB is one of the gatekeepers that is embedded in the host membrane. As expected in the HFQ mutant, both TIR and ESBB levels are also upregulated. And so it appears that HFQ seems to negatively regulate the expression of this regulatory operon, which is critical in virulence. And in many instances, when HFQ targets all the genes in an operon, what it does is it does this by binding to the 5' leader of the first gene in the operon. And so we inspected the region, the 5' UTR of grill R, and we saw that there was a recognizable binding site that is recognized by the distal surface of HFQ. So this suggested to us that maybe HFQ might directly bind to the grill RA mRNA to exert its effect. And to test this, we took the 5' UTR of grill R and about 45 nucleotides into the open reading frame, and we fused it to this truncated LAGZ gene that lacks its 5' UTR and start codon, right? So pretty much the translation of LAGZ is going to be under the control of the post-transcriptional regulatory elements of grill R. So the idea is simple, right? If HFQ is exerting its effect by directly binding to grill R, then now this hybrid grill R LAGZ mRNA should fall under the control of HFQ and it should be repressed by it. And we chose LAGZ because it provides really easy quantifiable means, right? LAGZ can take this colorless substrate ONPG, convert it to ONP, and you can monitor the color production. So what we saw was that in the HFQ mutant, there was increased beta-gal activity from the grill R LAGZ fusion, suggesting that that this cloned region of grill R was sufficient for HFQ-dependent regulation. Right? So in the wild-type strain, when HFQ was bound, we see very low beta-gal activity, and then when we deleted HFQ, there's nothing to repress it, and we ended up seeing much higher beta-gal activity. So. And so this helped us a lot because now we had a reporter strain that could allow us to identify potential HFQ-dependent small RNA, because we know this region of GRIL-R is sufficient for HFQ-dependent regulation. So the next step, what we did was to identify which HFQ-dependent small RNA could be potentially binding to GRIL-R and exerting its effect, we overexpressed each individual HFQ-dependent small RNA in the reporter GRIL-R lag-Z strain. So in yellow here, you're seeing this is the empty vector, and the blue line indicates a twofold reduction in the beta-gal activity. So we identified 3 small RNAs, MGRR, RIB, and MCAS, that appear to repress the beta-gal activity from this GRIL-R lag-Z fusion by more than twofold. And so up until this point is what we've published. Excuse me. And Marissa actually played a very, very important role in identifying these 3 small RNAs. And so the next piece, now all the data that I'm going to show you, this is all unpublished. And so we focused on MGRR, and we wanted to see how exactly MGRR is exerting its effect on GRIL-R. There is, when we align the 5' UTR of GRIL-R with that with MGRR, we see that there's a region of potential base pairing between the two, suggesting that MGRR may directly regulate GRIL-R. And so, as expected, when you overproduce MGRR, it base pairs, and you see this repression in beta-gal activity. And so to test for base pairing, what we did was we then mutated this trinucleotide sequence in MGRR and replaced it by GGA. Mutation of the idea here is that this mutant allele can no longer base pair, and you should no longer see repression. And this is what we see. There was partial restoration in beta-gal activity, consistent with the prediction that MGRR is base pairing to GRIL-R. We did a reciprocal experiment in which now we mutated GRIL-R. So we mutated this region. And then we wanted to transform it with this mutant allele of MGRR. This completely base pairs to this. So the idea would be, it should be able to repress the mutant. And this would genetically confirm direct base pairing between MGRR, small RNA, and the GRIL-R mRNA. However, even before we could do that experiment, the mere mutation, changing this GGA motif to CCA, it dramatically reduced beta-gal activity from this mutant fusion. Now, this was really surprising, but it did point to an important fact that somehow this trinucleotide motif is required for high levels of beta-gal activity. So when we looked at this, the trinucleotide sequence that we mutated, which was this, it's located immediately downstream of this dinucleotide AU. And as it turns out, these kind of motifs, AU-GGA, they actually serve as the binding site for this protein CSRA. And I would know that because I spent 5 years of my PhD actually working on the regulation by CSRA, but on a different gene. And so what we thought we'd done is we may have inadvertently mutated the binding site of CSRA in GRIL-R, and that's why we saw reduced expression. So this suggested to us that maybe what's happening is CSRA might serve as a positive regulator for GRIL-RA. And so to test this, what we did was we mutated CSRA in the GRIL-R lag-Z fusion, and as expected in the mutant, the beta-gal activity is dramatically reduced, corroborating our hypothesis that CSRA is a positive regulator of GRIL-R. And more importantly, in the mutant GRIL-R fusion, in which the CSRA binding site has been eliminated, when we mutated CSRA, we did not see any further reduction in beta-gal activity than what was already in the wild type. So this tells us that the transacting factor, the protein CSRA, and its binding site, here, they're part of the same pathway. Now, consistent with the positive regulatory role of CSRA on GRIL-R, CSRA also had a positive effect on GRIL-A, and when you mutated CSRA, we do not see any GRIL-A protein. And consistent with this, the CSRA mutant, it cannot form pedestals or attaching any facing lesions on infected cells. Here's the wild type strain. And all these spots that you see, those are actually pedestals that the bacterium is forming. So it's kind of like the bacterium sitting on top of, the infected host cell. CSRA mutant cannot form any of that. All right. So genetically, it appears that CSRA might be binding to the 5' UTR of GRIL-R. And so we wanted to verify this biochemically, right? So what we did was we generated an in vitro transcript that contains the 5' UTR of GRIL-R, which has the CSRA binding site, and we radioactively labeled it. And then we incubated it with purified CSRA protein. And if CSRA did bind to it, it would create a larger ribonucleoprotein complex, which would have reduced electromobility on a native gel. And that's exactly what we see. Here is U1 that represents the unbound transcript. And S1 and S2, they represent the shifted transcript, which is bound to the CSRA protein. So this showed us that clearly CSRA is directly binding to this GRIL-RA transcript. And so up until this point in time, so I hope I've been able to convince you that the RNA binding protein CSRA and the small RNA MGRR, they actually appear to bind to the same region in the 5' UTR of GRIL-R. And so this alluded to a potential mechanism by which MGRR may be exerting its effect. And it seemed to us that maybe what MGRR is doing is it's counteracting the effect of CSRA, right? So it's preventing CSRA from binding, and thereby it's exerting that's how it's exerting its effect. So to test this, we did a really simple experiment. We took that mutant of the GRIL-R LAGZ fusion where the CSRA binding site has been eliminated. And so if MGRR, if all that it's doing is preventing CSRA from binding, then when we overexpress this mutant allele of MGRR, which is completely complementary to this mutant allele of GRIL-R, so it can base pair, if all that it's doing is if it's preventing CSRA from binding, then we should not see any further repression by MGRR because the binding site for CSRA is already eliminated. CSRA is not going to bind. However, if MGRR is exerting its effect independently of CSRA, then we reason that we would see even more repression upon overexpression of the MGRR mutant allele in the GRIL-R mutant LAGZ fusion. So what we did see was that the MGRR mutant allele, it did not show further repression compared to the empty vector. Right? So this suggested to us that what MGRR is doing is it's likely all that it's doing is it's binding to the same region on GRIL-R where CSRA is binding and counteracting the effect of CSRA. We are currently doing some more biochemical studies to validate that. All right. So up until this point, I've shown you that MGRR appears to base pair to the 5' UTR of GRIL-R and it represses GRIL-R. But I haven't shown you any data on what MGRR is doing to the downstream gene GRIL-A. Right? So we reason that maybe MGRR is likely having the same effect on GRIL-A that it is on GRIL-R. And so when we did the experiment, I was pleasantly surprised because as it turns out, MGRR is actually having an opposite effect on GRIL-A. So overexpression of MGRR is activating the expression of GRIL-A, but overexpression of MGRR is downregulating the expression of GRIL-R. And this is not artifactual either because we assayed for the abundance of other proteins that are positively regulated by GRIL-A, right? And you'd expect if the GRIL-A levels are higher, layer levels would be higher, and so would ESBB. And that's exactly what we see, that overexpression of MGRR activates the downstream targets of GRIL-A. And so based upon this, based upon all the data that we have right now, it appears that what's going on is CSRA appears to bind to the 5' UTR of GRIL-R, and it stimulates the expression of both GRIL-R and GRIL-A. Now, it's been shown that you need almost twice the amount of GRIL-R, for it to be able to bind to and inactivate GRIL-A. And so we reason what's happening is that initially when the GRIL-R levels are low and GRIL-A, when most of it is free, it can activate LAIR, which in turn can activate transcription from the other operons and result in pedestal formation. Now, MGRR, and when GRIL-R levels become high enough, it'll bind to GRIL-A and inactivate it. When MGRR is overexpressed, MGRR is binding to the same region where CSRA is, and it's likely antagonizing the effect of CSRA. But MGRR only seems to downregulate the expression of GRIL-R, whereas it stimulates the expression of GRIL-A. And so you'd expect that when MGRR is overexpressed, there's going to be higher expression of these operons, and there's going to be much more pedestal formation. And while I haven't shown you this data, we've done qRT-PCRs, to validate the expression of representative genes from these operons. And that's what we see when you overexpress MGRR. There's increased expression here, and there's increased pedestal formation. And so this is really interesting because thus far, no one has identified the mechanism of action of MGRR. Excuse me. And we think that what MGRR is doing is its mechanism is, it's pretty much to counteract the effect of CSRA. And in fact, the most conserved region on MGRR is the region that it uses to base pair to its target, which is perfectly complementary to the CSRA binding site. Okay. Oh, sorry, folks, I'm just keeping track of time. All right. So that finishes the first story that I wanted to tell you guys. I want to tell you, a recent adventure that we've started taking. And this is really, really close to my heart because this project, it started off as a long-term, lab-based project for undergraduate students. So I teach this course at Emory, or not at Emory. I teach this course at St. Joe's. It's called Bacterial Pathogenesis, and it has a 14-week lab component where students go about identifying virulence factors in enteric pathogens. And so what they do is I give them, students are divided into groups of 4, and then I give them a list of 5 regulatory genes that are present in enteric pathogens that are related to EPEC. And then what students do is they have to extract the nucleotide sequence of those genes from those enteric pathogens using computational approaches. And then they have to identify whether the homologs of those genes are present in, EPEC, right? And if so, what is the extent of conservation? Based upon the genes that they know are conserved in EPEC, they predict which of those can possibly be involved in virulence of EPEC. And I'd be happy to talk to you about, the details of this conception if you're interested. And so once they've predicted a gene that could potentially contribute to the virulence of EPEC, then what they do is they generate mutants. And then they compare the phenotype of the wild type of the mutant to see if they affect any virulence associated pathways. And in fact, this is a result that I've taken from one of my undergraduate students, their lab project, where they predicted that RPON would be involved in the virulence of EPEC. They deleted it, and lo and behold, they saw that the RPON mutant doesn't produce any of this protein tier. So over the course of 6 or 7 years that I've taught this course, my students have identified a total of 13 novel discoveries for different for different genes that contribute to the virulence of EPEC and other related pathogens. In fact, one of my papers that I published in 2016 was pretty much a paper on the small RNA spot 42, and the initial discovery was made by students in this class. And this month itself, I'm resubmitting this paper that I'm going to talk to you now on OMAR-A and OMAR-B. And again, the initial discoveries were made by undergraduate students in this course. So OMAR-A and OMAR-B are 2 small regulatory RNAs, and they play an important role in modulating the composition of the outer membrane in gram-negative bacteria. Now, OMAR-A and OMAR-B, they are paralogs of each other, and they were arisen by gene duplication. So what my students did was they overexpressed OMAR-A and OMAR-B, and they assayed for the steady state protein levels of ESP-A, ESP-B, and TEER, all of which are involved in the virulence of EPEC. And as you can see, overexpression of these small RNAs completely abolishes the expression of ESP-A, ESP-B, or TEER. The top band is TEER, the bottom band is a non-specific band. So this suggested to us that OMAR-A and OMAR-B, they negatively regulate the Li and EPEC. And consistent with the previous results, the next step, what they did was they assayed for the expression from each of the Li operons. So they looked at representative genes highlighted in blue. And in each case, they saw that the expression of those genes was dramatically reduced. So this suggested to us that OMAR-A and OMAR-B globally shut down gene expression from the Li. And so now, they could only do this much. And so because they got all of this exciting result, I decided to pursue this further as a part of my lab. And so one of my undergraduate students who had taken that course, she joined my lab and she started to do further studies on this. And so she had predicted that because OMAR-A and OMAR-B are regulating all of these different genes in the Li, it's likely that they are exerting their effect through one of these two major regulators, GRIL-A or LAIR. And so what Sarah Mucci, what she did next was she assayed for the steady state transcript levels of both GRIL-A and GRIL-R in the OMAR-A-B overexpressors. And you can see the transcript levels were dramatically reduced. And consistent with this, the protein levels for GRIL-A was also practically abolished when you overexpress OMAR-A, OMAR-B. So this suggested that maybe OMAR-A and OMAR-B are targeting the GRIL-RA transcript. And so to test this, what Sarah did was she engineered a translational fusion in which she took the 5' UTR of GRIL-R and the first 45 nucleotides, right, and fused it to LAG-Z. And then she overproduced either OMAR-A or OMAR-B in them to see whether OMAR-A or OMAR-B could be targeting the 5' UTR. OMAR-A and OMAR-B really do not seem to be base pairing to the 5' UTR of GRIL-R, as evident from this. There wasn't much of a difference compared to the empty vector strain. So this suggested that it's likely that OMAR-A and OMAR-B are both exerting their effect on GRIL-R indirectly. And so to test, for the indirect effect, we wanted to see whether could it be that they were targeting LAIR, because there's actually this feedback loop between LAIR and GRIL-A. And so if they are not targeting GRIL-A, could it be that they are actually exerting their effect on GRIL-A indirectly through LAIR? And so to test that, first she assayed for the transcript levels of LAIR. As expected, overexpression of OMAR-A and OMAR-B dramatically reduced the LAIR levels. So this is the mRNA. Next, what she did was she looked at a transcriptional fusion. So here we have the LAIR promoter that is fused to, along with its 5' UTR and some of the N-terminal codons, it's fused to LAG-Z. But LAG-Z, it's under the control of its own 5' UTR, right? So this is a transcriptional fusion. Overexpression of OMAR-A or OMAR-B shut down beta-gal activity from this fusion. Right. So next, what Sarah did was she actually removed the native promoter of LAIR and replaced it with a heterologous promoter. So now we only have the 5' UTR of LAIR and the first 45 nucleotides to see if maybe OMAR-A and OMAR-B are directly base pairing to the 5' UTR of the LAIR mRNA. Surprisingly, we didn't see any effects of OMAR-A or OMAR-B. So this repression that we see in the transcriptional fusion, it's lost in the translational fusion, suggesting that you need the Li1 promoter. And because these small RNAs, they exert their effects by base pairing to target mRNAs, this suggested to us that the OMAR-A, OMAR-B repression of LAIR must be indirect. So there must be another regulator, which in turn is either it's activating LAIR, so they're repressing it, or they are activating another regulator, which is then repressing LAIR. And Currently, my lab is working to identify those regulators because we've tested 2 regulators that are known to be controlled by OMAR-A and OMAR-B, that CSGD and AMPAR, and neither of them seem to regulate the transcription of LAIR. So thus far, so what we have is we have these 2 paralogous small RNAs, OMAR-A and OMAR-B, both of which negatively regulate the LEE. And it appears that they globally shut down transcription of LEE by indirectly targeting the transcription of the master regulator LAIR. We know that the regulator through which they exert their effect, it's common to E. coli and EPEC, and it binds within 200 base pairs upstream of the transcription start site of LAIR, because a transcriptional fusion, it only contains the 200 base pairs upstream of the transcription start site. And so currently we are actively researching to identify the nature of this regulator, or the regulators through which OMAR-A and OMAR-B regulate, through which they exert their effects. So in the last, part of the, this talk, I would like to talk about the RNA-based landscape that controls the LEE and EPEC and therefore contributes to the virulence. So I hope I've been able to convince you that the small RNA, MGRR, it counteracts the effects of CSRA to affect gene expression from the LEE. And the other data that I showed you was that OMAR-A and OMAR-B indirectly affect transcription from the LEE1 operon of EPEC. Now, I haven't shown you other data that we've published, but my students have also identified other small RNAs, like we've shown SPOT42 represses the LEE through the TNA-CAB pathway. We've shown that this iron-responsive small RNA, RIB, also directly base pairs to GRIL-RA and shuts down the LEE. And then there's this nutrient stress-responsive small RNA, MCES, that hybridizes to GRIL-A. And most recently, a student in my lab has shown that this small RNA, DSRA, also indirectly targets the LEE by affecting transcription from LEE1. So with that, my lab has identified about 7 small RNAs that control virulence in EPEC, and I'd be happy to talk to you about this, outside of this. Now, in the last, Marissa, how much time do I have? I want to make sure I do not go over.
Yeah, you have about like 20 minutes, a little less.
I do? Wow.
Yeah, because we finished at 6:15. So yep.
Oh, but you don't want me to talk all the way up to 6:15?
Yeah, because I guess we can leave 10 minutes for questions. So probably like about 10 more minutes for the actual talk. Yeah.
All right. Sounds
Thanks,
good.
Dr. Bach.
So in the last part of this talk, what I want to do is I want to very quickly touch upon this pathogen called Escherichia albertii. And I want to compare the role that this RNA chaperone HFQ has in the virulence of EPEC and E. albertii. Now, I'm going to really quickly go over this stuff because in the beginning, it may not seem really interesting to you, and I'll get to that why. So E. albertii this pathogen was first isolated in 1991, and it was mistyped as Hafnia alvei. And its misidentification has persisted even to this day. In 2014, that was the first genome of this bacterium was sequenced, and in 2016, my lab developed the very first technique to engineer mutations in E. albertii. And in fact, it was Marissa who co-developed this technique with me. And so since then, we've been able to use E. albertii also as a model organism for my undergraduate students in bacterial pathogenesis. And so they've been knocking genes out in E. albertii and testing for their phenotypes. So again, thanks, Marissa. So here you can see that E. albertii is capable of forming pedestals, right? So these protrusions that you see, that is actually polymerized actin. And in red that you're seeing, that's tear that the bacterium has, transported into the host cell. And these blue dots that you see sitting on top of the pedestal, that is actually the bacterium. So we showed that E. albertii is capable of forming pedestals, just like EPEC is. This was important because there hadn't been much work done in E. albertii. Now, when I started studies on E. albertii, there was nothing known about the regulation of the Li in E. albertii. All that we knew was how the Li is controlled in EPEC and EHEC, right? And I've already shown you this circuit. So I was really interested in seeing how, if and how does the RNA chaperone HFQ control the regulation of the Li in E. albertii? But because we didn't even know the core regulation of the Li in E. albertii, we first needed to establish that. And so what we did was, students in my lab, again, undergraduate students, they knocked out LAIR to see whether LAIR was actually involved. And so when you knock LAIR out, there's reduced expression of this protein TIR, right? As expected, that reduced protein level is because of reduced mRNA levels of TIR. And so not only were the levels of TIR reduced in the LAIR mutant, but gene expression was globally affected from each of the operons of the Li. And you can see in each case, gene expression is reduced in the LAIR mutant. So this told us that in E. albertii, like in EPEC, LAIR is a positive regulator of the Li. And as expected, if you don't have LAIR, you're not going to express any of the genes from these operons. And so you shouldn't expect to see pedestals, and that's what we see. The LAIR mutant doesn't form any pedestals. So these structures, this is the wild type strain. You can see the wild type E. coli, wild type E. albertii is forming these pedestals, but the LAIR mutant doesn't form any. Next thing we did was we assayed for the role of GRIL-R and GRIL-A. Again, we generated a double mutant where we deleted both GRIL-R and GRIL-A. And as expected, in the double mutant, again, you don't have any TIR. And when we provided either GRIL-A, the activator, or GRIL-R, the repressor, only when you provide GRIL-A on a plasmid did we see that the TIR levels were restored. When we expressed GRIL-R, it was not restored, which is expected because GRIL-R, it exerts its effect by counteracting GRIL-A. So that makes sense. If you overexpress the downstream gene, it would restore the expression of that. And likewise, in the GRIL-RA mutant, the TIR protein levels were downregulated because the mRNA levels were downregulated in the mutant. And as seen with the LAIR mutant, gene expression was reduced from all of the Li operons, right? So this suggested that GRIL-RA, again, it's has a positive effect on the Li. Now, as expected, since GRIL-RA positively regulates LAIR, in this mutant, you don't have very little LAIR, and so you're not going to have much expression from these operons. And so again, you'd predict that there's reduced pedestal formation. And that's what we see in the GRIL-RA mutant. The mutant can barely form pedestals. You just see very few pedestals here. And they also have a really weird morphology. I can talk to you, later if you're interested in this. And so this suggested to us that, that LAIR and GRIL-R and GRIL-A have the same role in E. albertii as they do in EPEC. And so the next thing that we did was we could finally test to see whether HFQ, what is the role that HFQ has in E. albertii. When we deleted HFQ, we saw an increase in TIR protein levels. And likewise, this increase in protein levels were because of increased transcript levels in the HFQ mutant. And this was, again, a global effect where gene expression from all the Li operons was upregulated in the HFQ mutant. So this suggested to us that HFQ is a negative regulator, of the Li. And we've done further studies to demonstrate that, the way HFQ is exerting its effect. And in the interest of time, I'm just going to skip to the main point. Again, our data suggests that, as is the case in EPEC, in E. albertii too, HFQ appears to be targeting the 5' UTR of GRIL-R, through at least the small RNA MGRR. So all of this data paints a picture in which, oops, I'll just skip this, in which it appears that HFQ appears to regulate the Li in E. albertii in the same manner as it does in EPEC. All right, that's not really interesting. This is where it gets interesting. I showed you this data with OMAR-A and OMAR-B, right? The two small RNAs, when we overexpress them, it shuts off gene expression from the lead. This is what you see in EPEC. In E. albertii, though, the tier protein level, they are as it is, they're very low, and overexpression of OMAR-A or OMAR-B doesn't seem to repress it. In fact, we've done QRT-PCR too, and this is what we see, that overexpression of OMAR-A or OMAR-B in E. albertii does not repress the lead, but it only does it in EPEC. So even though these small RNAs are identically conserved in E. albertii, they don't seem to regulate the lead in the same manner in E. albertii as they do in EPEC. Another small RNA which doesn't seem to regulate the lead in E. albertii, but it does in EPEC, is this small RNA that I didn't talk about, spot42. So in EPEC, what spot42 does is it represses the production of indole. And we and other people had previously shown that what indole does, it can activate the master regulator layer, which can then turn on the other Lee operons and form pedestal. There's a really simple assay to detect for the presence of indole. You add this reagent called Kovach's reagent, and it turns red if there's indole present. Interestingly, E. albertii does not produce any indole, even though spot42 is identical in sequence in E. albertii as it is in EPEC. So at least we know that spot42, it's unlikely to regulate the Lee in the same manner in E. albertii as it does in EPEC. So what is interesting is, so we've come up with a model of how HFQ and HFQ-dependent small RNAs appear to regulate the Lee in EPEC. So we showed that, firstly, the master regulator layer activates the Lee in E. albertii, and then GRIL-R and GRIL-A have the same role in E. albertii as they do in EPEC, where GRIL-A is the activator, GRIL-R is the repressor, can and activate GRIL-A, and HFQ represses GRIL-RA. We have data that suggests that these small RNAs, MGRR and RIB, they have the same role in E. albertii as they do in EPEC. They base pair to GRIL-RA and regulate expression from it directly. In contrast, these small RNAs, which appear to regulate the Lee in EPEC, appear to have lost that regulatory role in E. albertii. So clearly, HFQ-dependent regulation in E. albertii, although similar, but it's not identical to that in EPEC. And so with that, I will end here. And I want to thank several of my former students. Many of you already know Marissa. Marissa has, she's done most of the work here. In fact, I don't think I would have been able to be as productive if it wasn't for Marissa. Here is Sarah. Sarah had worked on the OMAR-A, OMAR-B project. And here is Brian Critelli. He worked on DSRA, and we have a fascinating story with DSRA. And I'd be happy to talk to you about it some other time. And with that, I would be happy to take any questions.
Thanks so much, Dr. Bhatt. That was an amazing talk. I'm going to clap for everyone because that was just absolutely incredible. Okay, so Dr. Tam actually has a question. I can read it. It's also in the chat, Dr. Bhatt. He said, when are OMAR-A and OMAR-B expressed? Are they expressed more during biofilm formation? Since you're overexpressing these regulatory small RNAs, do you think that in the physiological expression of these small RNAs, they can also modulate the level of expression instead of a complete shutdown? I would think that one can look at the expression levels of OMAR-A and OMAR-B and the downstream targets in the existing RNA-seq data.
That is an excellent question. And, we are actually going to be doing the RNA sequencing experiment with a couple of the small RNA overexpressors, and actually OMAR-A and OMAR-B are, two of the four that we're going to be doing the RNA sequencing experiment with. So OMAR-A and OMAR-B, they are regulated by, the OMPAR-ENVZ two-component system. So, in response to, osmolarity can definitely affect the expression. Now, so one of the questions you said is, do you think that in the physiological expression of these small RNAs? Yeah, absolutely. So if I understand your question correct, do you mean whether they can have a tiering type of effect where there could be a graded expression of targets as opposed to complete abolition of the target gene? Is that yes, absolutely. That's what we actually think is going on with some small RNAs. That is what happens. You know, again, it depends on the level of expression of the small RNA. So yes, absolutely. So there's another question. What is the difference between typical and atypical EPEC? So a typical EPEC strain, this is one that contains the EPEC adherence factor plasmid. So atypical EPEC strains are those that do not contain the plasmid. So either they've been cured of that plasmid or, you know, they just don't have it. And the reason why, that is important is because the EPEC adherence factor plasmid, it contains the type 4 bundle forming pili, and it's also been implicated in the virulence. That's why there have been many reports that suggest that typical strains of EPEC tend to be more virulent than atypical. However, there have been other reports that suggest otherwise, that atypical strains have evolved other virulence-based mechanisms that can make them as virulent, if not more, than typical EPEC. And what's interesting is that, you know, we are actually seeing a shift in terms of what strains are routinely obtained from outbreaks. In the 1980s and 90s, most of the outbreaks that were associated with EPEC infections were because of typical EPEC strains. But in the past decade, there's been a switch, and most of the isolates that are being identified are actually atypical isolates, and there's actually much more heterogeneity amongst atypical isolates than there is between typical isolates, you know, within that group.
Yeah, thanks, Dr. Watt. And speaking of type 4 pili, actually, Dieter had a great question. He said, indole, I believe, also regulates the type 4 pili of EPEC. Does E. albertii
Yes.
have type 4 pili?
So indole does regulate the type 4 pili. E. albertii, actually, that's a really interesting question. I don't know if it has the type 4 pili. I know that it does. So if isolates of those have it, it'll have to be on the chromosome because it's been cured. You know, E. albertii does not possess the adherence factor plasmid. And in EPEC strains, it's located on the EPEC adherence factor plasmid, right? In fact, that was one of the reasons why, you know, E. albertii was routinely misclassified, because for a long time it was thought that it was an atypical strain of EPEC. But, you know, subsequent genetic and biochemical studies demonstrated that there are, you know, many more differences between EPEC and E. albertii. So it actually, E. albertii tends to be indole negative, most isolates, and it cannot utilize lactose, even though it has a functional LAC-Z gene. LAC-Y is non-functional. So, yeah.
Awesome. Thank you so much, Dr. Bott. That was an amazing talk. Round of applause again. And I know that you do have a few slides on your career trajectory. So if anyone wants to stay and hear that, I think that would be really neat, especially for the trainees here to hear about, you know, an academic position and how you got to your stage at St. Joe's. Yep. So if you want to share your those slides, Dr. Bott, you're more than welcome to.
Okay, Marissa, I promise I'm not going to take too much time.
No, yeah, this is great. Yeah, thank you.
And this is great. We have a lot of students that are interested in an undergraduate career path and combining research and education. So you're a great role model for many of our students. So thank you so much for doing this for us.
And Marissa, of course, you know, if anyone wants to also just email me or just, you know, about any question, please just, Marissa, you can just I'll give you my information too, but, you know, you can also give it to them, right?
Thanks, Dr. Bott.
I'd love to talk to you guys about this in more detail.
Absolutely.
Because I know how confused I was actually in undergrad and even in grad school. But, you know, it was my mentors who really helped me out. So here, let me share my screen.
Thank you, Dr. Bott.
Well, thank you. Okay. So Marissa, can you see my screen?
Yep, it looks great.
Okay, so I think it's just 4 or 5 slides, not too many. So I actually, I grew up in a really small hill station in India. It was Nainital, India. I grew up at the foothills of the Himalayas. And it was there that I really, loved biology because, I was surrounded by nature. I would go on long hikes with my uncles and I loved learning about, even simple things like, it started off with love for plants, like, with transpiration, with things like that. So I went to a boarding school there, and there, my mentors really helped me hone my love for both chemistry and biology. And so I cannot thank them enough. But the thing is, I even while I was growing up, I wanted to be a doctor. And the reason is why my family is flooded with doctors. And I really should not stereotype this, but I think it's typical of a typical Indian family, I guess. You have doctor, doctor, engineer, doctor, doctor, engineer. My family is like that. In fact, right now, there are 4 people in my household. My mom and my aunt are visiting, and my wife is here too right now. She works in New York. They're all doctors, MDs, and the PhD. So you can see, so here's my aunt actually. And I wanted to be an MD growing up. And I came to the US to pursue my undergraduate education because my mom was, she was posted in Nepal, but eventually she was going to be in the US. So I wanted to be here close to her. And so I went to Denison University. It's a really small liberal arts college in Ohio. That's where I knew that really completely altered my way of, looking at things and at what I wanted to really do in life. Up until this point in time, I had just by default equated biology to medicine. When I took my chemistry courses, my biology, especially my microbiology course, that's when I knew what I really, really liked was understanding molecular mechanisms. They were like jigsaw puzzles to me. I was fascinated by them. I just couldn't stop, I just wanted to do them. I loved it. I love biochemical pathways. I love metabolic pathways. And I knew that I'm more interested in understanding the molecular basis of phenotypes of phenomena. And so to give you an example, I told my mom that, I don't think you want me to be a doctor and to be an MD. And the reason why I knew why was because I wanted to see how a person's disease condition is exacerbated. I wanted to see what happens. You don't want me to be your doctor because instead of treating you, I would be more interested in seeing how your condition is worsened so I can study the underlying basis. You don't want a doctor like me. I told my mom straight up, I cannot be a doctor. You don't want a doctor, because. And so from there, I knew, I really liked research. I had the opportunity to do research. And I also was asked to TA for my molecular biology course. And that's where I fell in love with teaching because it was kind of like, there was this one time I remember that I was explaining a process to a student who was a semester older than me. And he actually said this, "Oh, you're right." And that was, it was the kind of like that the feeling of joy that you get. It's unparalleled. No kind of monetary satisfaction can parallel the joy that you see in a student's eyes when they've answered the question or when they grasp that thing. It brings you so much of satisfaction. And for those of you who've had those instances, everyone here probably has had those, and you could probably relate to that. So that's when I knew I just wanted to continue experiencing this joy for the rest of my life. And I figured teaching is what it is. And so I married my two loves of, teaching and doing research. And I knew I wanted to do something that merged teaching and research. And so from there, I decided to, get my PhD at Emory. But I knew that I wanted to do teaching and research, but I didn't know where I wanted to go. Would it be a big university, a small university, or what? It was at Emory that I had the opportunity to teach some more classes while doing my own research. And I saw the pros and the cons to being at a big research intensive university like Emory and at a small liberal arts college at Denison, where I love that experience. It was a more intimate experience where class sizes were small. You could interact with your mentors more closely. There were both advantages and disadvantages of both. And the former, my experience in Denison really made me reflect a lot more about the direction I wanted to go. And so that's when I decided that I really want to end up at a small liberal arts college like Denison. And so, I did my postdoc at the NIH. I worked on small RNAs there. And after a few years, I married again my pre-doctoral research on EPEC with my postdoctoral research on small RNAs. And so that's what I research now. I look at the role of small RNAs in the virulence of enteric pathogens, and pretty much I do it all with undergraduate students. Well, most of them undergraduate students. There are limitations to a place like St. Joe's where the infrastructure is limited. Some of the tools and technologies are limited, not as advanced as you have at Penn. However, I get to also do what I really love besides research, and that is teach. I teach like 3 courses each semester besides doing research. And it provides a beautiful complement to me that, educating the next generation of scientists and doctors, while at the same time, I'm also able to intellectually stimulate myself by continuing to do research. So, yeah, so that's.
Thanks, Dr. Bott, for sharing your story.
Yeah,
it's so inspiring. And I'm very grateful to have experienced your mentorship firsthand. So thank you so much.
Oh, thank you, Marissa. Thank you for all the work that you've done in my lab and for, for inviting me to give this talk too. I cannot thank you enough.
Oh, we can't thank you enough. You're the perfect speaker, perfect person for this talk. So thank you so much again. If anyone has any questions, I'm sure they can definitely reach out or ask now.
What if I ask a quick question, Marissa?
Yeah, of course.
So I'm Michelle. I'm the student chapter advisor, and I one of the things that you just said really resonated with me, and I get a lot of questions from students about when they are doing their postdoctoral fellowship and then choosing an area of research that they can take from their fellowship that then becomes their own independent project, and how that conversation with your postdoctoral mentor, how important it is to talk as early as possible about, I want to have my own lab, I want to develop independent research. And how did that go for you? You were at the NIH and you developed, it sounds like you combined your 2 areas of interest, the RNA work that you did at the NIH with your previous microbiology project.
Right. So when I interviewed at the NIH, so I did my postdoctoral work in Gigi Storrs' lab. And when I interviewed with Gigi, I had the opportunity to talk with her postdoctoral fellows, others who were already there. And it was conversations with them that I asked them, like, look, how is Gigi as a mentor, and what kind of projects are you working on? Will Gigi allow you to take some aspects of these projects with you when you go to your lab? And every single person that I talked to had said, yes. Gigi said that, you can take this. They talked about it. And so that made me feel a lot more comfortable knowing that at least some aspect of this I would be able to take with them. But then even before I joined the lab there, I talked with Gigi because I wanted to hear this from her, and asked her, like, Gigi, will I be able to take aspects of this project with me? She's like, yeah. And Gigi she is such not only a remarkable scientist, but she has supported me so much through so many years of, ups and downs. I cannot thank that woman enough. I mean, the things that Gigi has done is just, oh my goodness, I can just sing millions and millions of praises of her. She's amazing. Yeah, she goes way out of her way to help, regardless of whether it's a graduate student, whether it's a postdoctoral researcher. She's many researchers are oftentimes, they want their prodigies to follow in their footsteps. Like, if you're in an R1 institute, you need to go to an R1 institute. Gigi's not like that. She helps you walk through the door that you are interested in, not what, she wants you to do. So that's, I think it's really important having supportive mentors throughout your, academic trajectory. Otherwise, it can be, sometimes the consequences can be really disastrous. So
it sounds like you're carrying the torch forward.
So pay it forward, right? So
that's right.
Yeah.
Great. All right, great. Yeah, does anyone else have any more questions for Dr. Bott? I have a quick question, actually, Dr. Bott. I think I know the answer to, I do know the answer to this, but I think it might be helpful for other people to know. So what kind of teaching experience did you get in grad school and then also as a postdoc to kind of prepare you for a professor role at St. Joe's?
So again, I think it's really important that so it's important that your PI be comfortable with you teaching, because if you know that teaching is something that's very important to you and your PI is reluctant to have you teach, then again, you may want to think what lab you want to be in. But for me, both my pre-doctoral advisor and my postdoctoral advisors, Gigi, they were very, very supportive. And as a pre-doctoral student, when I was getting my PhD at Emory, I TA'd medical microbiology. I would also tutor at, for organic chemistry. I loved organic chemistry, so I would do that. I would teach a couple of times at a high school. So any kind of experience actually matters. If you've even mentored at, like if you're mentoring an undergraduate student in your lab, right? Mentoring is a form of teaching, right? It's more intimate one-on-one interaction. All of that goes a long way into showing that you are interested in teaching. But again, there are many opportunities that universities provide, right? Especially big universities. Like I believe Penn has the Penn Port program. Emory had, what was that? I forget. The FACES experience. So there are lots of opportunities because they all, they're always looking for TAs or SIs. And in big universities, oftentimes the TAs are going to be your graduate students, right? So there are lots of opportunities to be able to do that. At the NIH, they actually have a whole wing that's actually dedicated to curriculum development too. I think it's phase, that it escapes my mind, but they actually they teach many different courses, and they encourage you to build your own curriculum. You have workshops for developing curriculum. So it provided me lots of tools to be able to hone my craft, and I continue to improve on that just because of some of the guidance that I received when I was at the NIH.
That's awesome. Thank you, Dr. Bott, for sharing your story.
Thank you, Marissa.
Really appreciate it. Great. Okay, if we don't have any more questions, then I think we'll wrap up for today. But thank you to all of our speakers, and thank you especially to you, Dr. Bott, for staying and sharing, your journey to academia. It's really, really incredible, and we're very grateful. I'm personally very grateful to have experienced your mentorship, and I'm also very thankful to have you here today. So thank you.
Thank you, Marissa.
Yeah, and with that, I think we'll wrap up.
Great job to the student chapter officers. Great job today. So
Absolutely.
and thanks for your work and planning.